good morning to all of you. Thanks for being here. It's a real pleasure to have Elizabeth being here today giving a seminar. Yeah, I know Elizabeth since quite a while. We're working on NucNet together. Maybe some of you heard that's a big international project that got initialized in 2007, I think, and has by now over 170 sites.
171
sites. So I think Elizabeth will tell us quite a bit about that. But where does Elizabeth come from? From Minnesota at the moment, but she's in Netherlands at NEO in Wageningen for sabbatical, so it was closer, but the flight got cancelled anyway yesterday. But Elizabeth has a bachelor from Ohio in biology. I was surprised to see the religious study minor, but that was mostly history, which is pretty cool. And then she went on to California for a PhD at the University of Santa Barbara. But in between, I also learned this morning, and actually would have been here, she worked as a construction worker and quite a lot of different things. And after the PhD, she stayed in California at Berkeley, was a postdoc there. And there was another postdoc also out in California. But then she moved to Oregon State as an assistant professor and also associate professor for a while. And in 2017 moved to Minnesota, where she currently is now a professor, but also has two other functions. One is just an endowed chair for microbial ecology, which is new for the next 5 years, which is pretty cool because a lot of new work will be done, I'm sure. And she's also the distinguished professor for her career awarded last year as well in Minnesota. You will hear what Elizabeth is doing, but she has a very broad scope going from botany to microbes to soils, the other things. So very happy to have you here. Floor is yours.
Yeah. Great. I gather there are some people here. Hi. Maybe or maybe not. That's where the religious studies comes in. It's really fun to be here. It's super fun to put humans with either people I've talked to on Zoom or just names in the literature. So that's really fun. And of course, to catch up and talk about science. So thanks for having me. Today I'm going to talk about nutrient network and some of the work that we've found about the relationships between consumers and the plants that they're, consuming. But I'll start. Oh yeah, great. Maybe. Okay. I don't know what happened there. Okay. What did you push when you were over here?
Got it.
Forgot talk. Okay, sorry. We're gonna try again. Oh, I can't even escape out of here. I need to stop. So, no. All right, I pushed escape and it moved to the next slide. So we'll see how this talk goes anyway. This will be something we'll all find out. So I want to start at the really big scale. Stepping back from Earth, looking at Earth with stitched together images from outer space. And what I see when I look at that is banding patterns of vegetation, right? So we see dark greens and lights, right? So this chromatic scale looking from space back to Earth. And I'm fascinated by what controls that. And, if you were to ask me from just this image, the answer would be, precipitation, of course, right? We'll see if this works. Let's see. Oh my God. But if you stitch together those same images through both space and time, there's a more nuanced answer that emerges, right? So when we look at the shifting vegetation banding patterns, we see that there's both temperature and precipitation that underlies those, right? And we're changing those radically. And so we're seeing that through human activities, we're radically changing that baseline on which climate, temperature, and precipitation, sorry, I'm not very good at this online camera thing, are changing. So we do see that same uptake and release annually, but on a background of changing climate. And it's impacting all of Earth's biomes, right? This isn't just restricted to one. We're seeing changes in all kinds of characteristics, wetter places, drier places than historical averages. We're seeing changes in species composition. We're seeing droughts that are unprecedented and fires that are emerging from that, and so on and so on. But I'm going to focus in on grasslands for a bunch of reasons. One is they're just beautiful. The other is that they're on every continent on Earth. And if you want to talk about Antarctica and tell me that I'm wrong, we'll have a conversation because we can all cry about this. There is one that's emerging in Antarctica now. They're also incredibly important for human history and human evolution. All of our food, well, not all of our food, much of our food, much of what you had for breakfast and possibly what you had in your, for your beer last night comes from grasslands, right? Grasses are a huge source of our nutrition. And so we have this very tight link with grasslands as humans on Earth. Oh, there we go. It's there's some weird delay here. But what are the kinds of changes? Right? We have enormous changes that aren't just climate, that are happening in grasslands around the world. So we have this huge increase in fertilizer production that is, for example, this is one nitrogen plant, but we have 400% increase in available nitrogen on Earth since 1950, right? That's not that long ago, I tell my students. But we're also shifting, you know, at the same time, but in different locations and at different rates, we're shifting what the herbivores are that are present. And so this is one example of cattle displacing elk. But the same thing is happening on every continent where we have a narrow range of herbivores, sheep, for example, sheep, cows, yaks, but a very narrow range of herbivores that are displacing globally diverse herbivores. And what is changing in that interaction is something that we haven't really well defined. Okay, so we have a lot of things that are happening all at the same time, but in different locations, right? So we have globally changing nitrogen coming in, we have changes in the density and identity of herbivores around the world, where we're expanding grazing into areas that are formerly forested, and so on, right? So we have many different changes that are happening all on top of, or maybe underneath, climate also changing. And so what will our future grasslands look like and how will they function is what drives me to start asking questions. So I ask, you know, how will these grasslands respond to, for example, increased nitrogen supply or nutrient supply, changing herbivore composition? And that's a really, really big question. So I want to take just a moment to think about how do I, as an ecologist, answer that question? I go to the field, right? I go outside and I ask the plants what they think, right? So I dump fertilizer on or I put up a fence. Like this experiment here, it's a little bit hard to see with the light, sorry about that, but you can see I have fences here. Unfortunately, well, you can kind of, if you, maybe, you can see this plot here, for example, is much richer, it's greener, there's a lot more plant, sorry you guys, it's okay, don't worry about it. There's a lot more plant mass in this plot. You can just believe me that that's what it looks like. And there's, you know, if I go and I crawl into that plot, I find that there's maybe a different composition of plants there than over here. And I compare those two, right? So I have the treatment and the control, and I say, okay, that's what the future will look like, right? Under increasing nutrients coming into the system, that's, you know, it will have more mass, we'll have fewer species, whatever we might have, whatever I find in that plot, and I compare it to control, and then I infer, and I start to worry. What can I know? And the biggest concern that I have in that type of study is, can we scale up? Can I know something from my plot here, and can I say something about my field? I feel pretty confident that I can do that. You know, I could probably tell you something about what would happen in the future in some other portion of my field if I did the same thing. And then I go to the scale of my state. This is the state of Minnesota. Yes, just for comparison, I was trying to figure out, working in the Netherlands, just scale is very different in Europe. If I laid Minnesota over Europe, it would reach from Amsterdam to Switzerland. And it's not a big state in the US, right? So that just gives you some sense of scale here. And can I say something about what would happen in another part of Minnesota? I don't really know. I start to feel really worried that I might not be able to. And just for scale there, I did a little exercise to figure out how many Switzerlands would fit into Minnesota. So it doesn't have the same relief, so I didn't take that into account. Minnesota is relatively recently glaciated, unlike the mountainous regions here. But nonetheless, can we scale up? Can we, if I go to Washington, DC, and my senator asks me, what can we say about the US, the 48 states, or North America? I don't know. And I feel like while we write with confidence in the discussion of our papers about the inference we can draw, I worry about that. And so we have tools. This is not something where I'm bashing ecology in any way. We have multiple tools. Ecological theory is a really powerful one where we can use math, a language of logic, and we can make inference. We can say, well, what would we expect? And does the biology agree with that? And if we get it wrong, if the biology doesn't agree with that, we need to go back to the math and think about what assumptions are we making that are wrong, right? And that we can mutually inform our understanding through that interplay. We have meta-analysis, which is another really wonderful tool. Studies can be done in Finland and Argentina, and I don't know where everybody's from this room, but they can be done and brought together, and we can ask, do we get the same answer or not? But there are challenges with meta-analysis because everybody in this room does their study a different way. And so the sorting apart the biological from methodological inference that we can draw is super tricky with meta-analysis. It's, from my perspective, a wonderful way to set up hypotheses, but not a great way to test hypotheses. But again, it's another tool in our toolbox. Okay. I'm going to tell you more about what worries me a little bit when I think about this problem of can we scale up, which is I talked about globally changing climate and, we've got elevated temperatures and changing precipitation, right? But we actually have very different things happening in different locations. We have a different combination of changes in herbivory, changes in nutrients coming into the ecosystem, changes in precipitation and changes in temperature for any one point. So if I do my work here, in Minnesota, what can I say about what would happen in, for example, Central Australia or India, where the conditions are absolutely different and the changes that are happening are completely different? We have a different future that we're looking at, and it's overlaid on a different history, evolutionary history, the geologic history. And so why would I even expect that I could say something from my site in Minnesota about the rest of the world? And so what we really need to do is do an experiment everywhere under all the conditions. And this was the dream that we had in, 2006, and then it started in 2007. It's turned into something much larger than any of us had expected. And I think that's because a lot of us wrestle with those same questions and the realization that, this is something that's achievable. It's not expensive, it's achievable, we can each do it. In the US, at the time that we started this, there was a huge amount of money going into this one sensor network. And I think it disempowered a lot of scientists because they're like, I can't do big science without big money. We're like, here's an alternative, right? So it's kind of game changing in this sense. And I think that's why we've had so many incredible scientists that really want to answer these questions add in and say, I'll do it at my site too, right? You guys have it just outside. Oh, wrong window. So this is the nutrient network. And I guess actually back here, one of the things that I do when I look at this map, the first thing I do is I say, oh my God, that's a lot of science. That's so cool. And the next thing I do is I go, look at all those places where there aren't sites. How come there aren't sites there? What can you know about the world if you're not sampling all the sites in the world, if you haven't collected the whole set? And so to allay my own concerns, I did an exercise, which was to graph out what is the range of combinations of different conditions that we're capturing with this data set. So while we don't have a dot in every place, have we captured broad gradients of climate, of soil chemistry, of productivity? And the answer is yes. So this red circle at the bottom, I'm just going to point out it here, this red circle indicates the range of values that we've captured within our data set for precipitation, for temperature, and for biomass, which is most of where plants are on Earth. So we've done a pretty good job while we don't have dots everywhere. I would love to collect the whole set. If you happen to know people that are working in, where there are plants growing, there aren't dots, let me know. But I do feel like we have pretty strong ability for inference. In numbers, we have just over 170 sites that span, well, almost 30 countries, 6 continents, and they go from Tierra del Fuego all the way to Svalbard in Norway. So it's really pretty much the extent of, almost the extent of grasslands on Earth. Sites have added in through time. And so we have at any one site between 1 and 15 years of continuous data where we've resampled through time. We have uniform data. If you've ever done one releve or quadrat, you'll know what this means, but we have more than 4,000 of these around the world. So that's kind of incredible. And we have, I don't think there's a direct correspondence between the number of surveys that we have done and the number of plants, although species area and time relationships suggest that we'll continue to collect them until we've collected the whole set. But we have about 1.5% of all of Earth's known non-tree vascular flora, which to me is astounding because it's not just where are they, like a flora would tell you, but what are the conditions under which they're persisting? So we're able to understand more about their ecology and not just where they are on a map. So I find that incredibly exciting. And we have discovered that we are in fact working in grasslands, so we have about almost 5% of all of known Poaceae. So it's a data set that's quite rich and allows us to ask a lot of questions. Okay, so what are these data I keep talking about? We have 3 different data types, 3 different data sets that we're collecting as a group. The first one is observational, just go out and collect data. And we thought that this would just sort of set a baseline. This would be a great place to, sort of start doing some comparative work. Those data have been incredibly valuable. So every site has 1 year of observational data, even before they do any experimental work. So we have observational data from a huge number of locations and have been able to tackle a lot of new questions with that. We have a multiple nutrient experiment where we have nitrogen, phosphorus, potassium. Well, potassium and micronutrients are one treatment. And that's added in a factorial combination, so 3 different treatments. And that gives you 8 plots, and then we have 2 additional plots where we take advantage of the all nutrient and no nutrient treatments. And we have fences around all nutrient and no nutrient, which gives us a whole new experiment where we have nutrients by fences in, again, a factorial combination. So these are 3 different data sets that have allowed us to tackle all kinds of questions that have been mostly theoretically motivated. Okay, what are the data? So you know the experimental design broadly. Oh, and each of those is replicated 3 times within a site. So it's minimal replication within a site. And the idea is that we have to do a frame shift as a field ecologist to think about a site as a replicate now, and not the replication within the site being the key to understanding variance. The core variables that everyone's collecting using the same methods everywhere are the cover of each species within a 1 meter area that we go back to every single year. So we sample exactly the same location through time, live plant mass and litter mass, so dead plant mass. Light interception, so the difference between above the canopy and at ground level. Plant provenance, which is a really cool data set. I'd be happy to talk more about that if you're into invasive species. It uses site knowledge of what is native and what is non-native to overlay that onto floras, for example. So it's an improvement over a lot of the flora data. And then soil texture and chemistry through time. Not every year, we have texture one time, but then we have chemistry approximately every 3 to 5 years from every site. Okay, so what are we doing with all these data? We're tackling these complex webs of interactions to try to understand questions like what I posed at the beginning. So do herbivores or fertilization, for example, control plant function, control plant diversity? And so I want to talk about those interactions. You can imagine, for example, that multiple nutrients might increase biomass, right? We fertilize, we get more plants. Like we've known that with agriculture for a really, really long time. But that biomass might change, for example, the relative cover of different species. Maybe they compete differently for that. It might change the amount and the chemistry of the litter, for example, which might feed back to change biomass. You can imagine again that more biomass, maybe less light, maybe not. Right? And that might feed over, these interactions might feed over to alter plant diversity. But if the herbivores are consuming, right, it's complex webs. We want to understand not just what's the answer, but where is the answer different? And that's what we can do with this design. So we can start to say under certain conditions, we get certain answers. Okay, but we have a lot of data. And so for me, I feel like I don't want to just sit down with the data and start, doing correlations or something because it's too much. There's too much information here. And so for me, I feel like starting with hypotheses, starting with math models is a really powerful way to begin. And so we started, I'm just going to give you one example. Here's the idea that in a world with just plants, we'd have an awful lot of plant. But if you put some herbivores in there and they eat the plants, you get more herbivores and less plants, right? Very simple. But that's where we start with math models, right? Where I start. This is laid out really nicely in a handful of papers. I just pulled one image from an Oxen and Nadal paper in 2000. Along a gradient of productivity, you see, you can ignore the carnivores here, but you see at very low productivity, there's just enough resources to grow some plant, a little bit of plant. That plant grows up to a point where suddenly it can support, there's enough plant coming into that system to support herbivores that are consuming it. So then we have persistence of what they've called folivores. If you continued this slope of the plants out, that's all of the offtake that's happening in this math model and turning into these folivores. And the reason that it's not the same slope is there's a conversion efficiency, right? The herbivores aren't taking all of that mass. Nonetheless, what we see is where the folivores are present, there's no change in biomass with increasing productivity. So it decouples the concept of productivity and biomass. Okay, so, oops, yes. So how would we test this? We would test this using this relationship. So control and nutrient, right? We're increasing the productivity of that system by putting more nutrients in the system and live biomass on the y-axis. If the herbivores are eating all of that biomass, we would see no change in biomass across this gradient, right? That's the prediction from math theory. But if we put up a fence, we would see more biomass, right? That's that same prediction from the theory I just showed you on the last slide. And we would see a smaller difference here with control and a much larger difference in the treatment, the nutrient treatment, because of that difference in what the herbivores are doing, right? So that's the difference in consumption. Let's just put that into log space. And there's a reason I'm going here. We go into log space. All I did was I took those numbers out of R and I stuck them, I just put them into log space. Because what we want to do is deal with variance differences among sites and use log ratios instead of just straight differences. If we move into log space here, it looks very familiar. This no change across control and nutrient outside of fences where herbivores are consuming, we would see as this nutrient, no change, right? There's no difference between those two values here and here. With the fence, you see an increase, right? From no nutrients to, no nutrients to nutrients added, right? And that is this value. It's greater than 0. And then these two arrows, the difference between these arrows, right? We see this is a much larger difference with nutrients added than control. And that's this interaction here, nutrient by fence. And there's a reason I walk slowly through that, because you're going to see a lot of these graphs. I find them incredibly informative. But there are other options. So that first one, that I don't know what just happened. For those of you online, I don't know.
Oh, the camera is still on, yeah.
All right, we're all good?
It looks like.
Okay. Right, so if we have, so that's the herbivores keeping up, right? They're eating all of the extra mass that's being produced across that gradient. But if the herbivores don't keep up, they're eating more, but they're not keeping up, we see a relationship that would look more like this in the field, right? So there's more mass, it's just some of it's getting eaten. If we put that into log space and over into these graphs that I was showing you, this is what it looks like. Focusing in here, these arrows, the difference is the same. Those two arrow lengths are the same in log space. That's what gives you 0. It's not that there's no change. It's just that the change is the same. Or if they're not keeping up, for example, in this bottom one, then you can see this interaction such that there's more offtake under control conditions, right? There's so much more biomass and there's just not enough herbivores. Maybe they're being controlled by something else, right? It's not food limiting. And so we would see this dot dropping below the line. Okay, so, all right, I don't know what you did, this doesn't work anymore. What was that? Okay, sorry. Nope. Nope. Okay, we tested this in meta-analysis. I don't know, I might have to keep walking over there. We tested this using meta-analysis. This was Dan Gruner led this across different ecosystems. And what we found was, in terms of the interaction, right, our herbivores keeping up, we found that they somewhat do in marine systems, but not so much in terrestrial. But the thing that we found, from my perspective, that was incredibly interesting here was that there were only 4% of all the studies we were able to find in the entire literature that were in terrestrial systems. And only 4 of those had vertebrate herbivores, which was not what I was expecting. And so that was the genesis of Nutrient Network, was realizing that these ideas just simply hadn't been tested despite their importance in how we think about how systems are regulated. So now, moving into NetNet, we have a lot of data across climate, geography, diversity, soils, invasion, right? And so we can now start to understand not just what's the mean, but with the variance, we can start to ascribe that variance to known measured factors. So we can start to understand the conditions under which herbivores are and nutrients may interact. So I'm going to focus in on that third data set that I talked about, the nutrient by herbivore data set. Okay, talking about things that worried me. At the outset of this experiment, I didn't tell you guys this before you started. One of the things that worried me most was I had just convinced a whole bunch of people around the world to put up fences, which were expensive and painful to put up, frankly. And, you know, fun on if you like that kind of thing, but hard to do. Yet, we have different herbivores everywhere, and they have different diets and different body sizes and different densities. And I'm just like, oh God, with all of that noise, maybe we won't find anything, right? We might not be able to ascribe it because I don't have replicates of kangaroos in South America or, you know, guinea pigs in Africa. It just, you know, we don't have that same replication that we do across, for example, climate. But there was an effect. So when I made this graph for the first time, I had a huge sigh of relief that in fact, there is a change in biomass across fence lines. So there's more biomass inside of fences. So that was a big relief. Even though you might say, well, of course, it wasn't a given. When we add nutrients, we do in fact find more plants growing. So that's also a relief, intuitive. But this was really what this experiment was set up to test. Herbivores are really not keeping up. That's what we're seeing, is that across the world, when we expanded out that data set, we found that no, they're not keeping up. And in fact, it looks like they're actually becoming less efficient. And I have a hypothesis I'm ready to test. So this is through time, 2 to 4, 5 to 7, and 8 to 10 years, we're seeing a decline in that maybe. They're still crossing zero, a decline in the efficiency of herbivores, which may be because of turnover toward less palatable plants. So that would be my hypothesis. We'll test it when we pull in the 11 to 15 year data set, which we can do fairly soon. I'm on sabbatical. So no, they're not keeping up, but in the sites where we also have, so we had wild and domestic herbivores, in those sites, they do keep up. So to me, what that tells me is they're not in sites where the density of herbivores is not limited strictly by the food at the site, what's available, the wild animals, right? These are subsidized herbivores. These are herbivores that are getting additional food. They're getting vet bills paid. They're getting their inoculations and their dips or whatever they get, right? They're able to keep up. So that also is very interesting to me. Where, right? So now all this orange up at, and Nadia, sorry. The orange at the top is highlighting what we couldn't have known if we hadn't repeated this experiment in a whole lot of places. So what we found is across a gradient of soil nitrogen, for example, and I'm just plotting the difference from control. That's what each of these lines is. Across a gradient of nitrogen at low soil nitrogen, when we add nutrients, we get more biomass, whether or not there are herbivores there. Those plants are limited by nutrients, and we add those nutrients, and even if the herbivores are there, they're not having as much of an effect. But at high soil nitrogen, at these sites, we find that fences are the biggest limitation, right? So it's these high soil nitrogen, highly probably fertile sites where we see the herbivores having the biggest effect. Also across mean annual precipitation, right? So each of these, again, is a site. So what we find is across mean annual precipitation at very low precipitation sites, we tend to see this largest effect of herbivores. And we don't see that as much. The herbivores are not having as much of an effect at these high precipitation sites, right? Maybe there's rapid regrowth, for example. What else is happening? And so scratching my head and staring at all of the results that we've had in Nutrient Network, one of the things I realized is from work that Michael Anderson led, on the x-axis here is precipitation again. This, let's see, in the solid circles, you can see what happens inside a fence. 2 on the y-axis is the fertilizer effect on biomass nutrients, so biomass chemistry. What you find is that at low precipitation sites, when you add fertilizer, you get really high nutrient plants, right? They're probably not able to pair it with carbons, right, to grow because they don't have enough water, right? Those are the areas where the herbivores are having the greatest impact, those high nutrient plants. And so it's likely that it's through that nutrient flux, that nutrient change in the chemistry of the plants, that we're seeing this large effect of herbivores in low precipitation sites. This then moves through to the ecosystem scale. I'm sorry, Pedro, I took out the slide on litter because I have too much here. But we can trace this down to the soils as well. This is work led by Judith Sitters. Anita's wondering why the name's there, but you'll find out in a sec. This is work led by Judith Sitters, where she found that inside of fences where they've been fertilized, you see actually a decline in soil carbon and a decline in soil nitrogen. And likely what I think is going on there is a change in the microbial communities themselves. So a hypothesis to test, it opens a lot of questions. But what I think is cool, and why I put Anita's 2 papers here, even though I'm not showing you data from those, is Anita's been working on some of the fluxes and thinking about the nitrogen fluxes in the microbial communities themselves. And I think our next step would be to try to bring these results together and really try to understand how the microbes are changing and how the soil chemistry is responding across this. So I think there's some of the results do and do not map onto each other, and I want to understand why. And I think that's one of the mysteries we need to keep, move in that direction, thinking about that. Okay, so what about plant diversity, right? There are a whole lot of other things than ecosystems, I know, it's hard to imagine. But we care about diversity on Earth, and so we can ask about that. What's the effect on diversity? Again, nutrients, fence, and that nutrient fence combination showing you that same style of plot, in this case, looking at diversity responses. We see that when we add nutrients, we lose diversity. But there's no mean effect of fences. And I have to say, this was actually the first plot that I made. And I was like, oh gosh, I'm in trouble, right? There's no mean effect of fences on diversity around the world, nor is there an apparent interaction. What's interesting is when we looked at which species are being lost, we found it's the native species in general. We're working now to understand whether that maps onto traits, for example, stature of plants. Are we losing just the small statured plants or not? But with our covariates, we were able to pull out something that I find really cool, which is where we take out where we take out herbivores, and that impacts light. So when you take out herbivores and it gets dark underneath the vegetation, those are the sites where we're losing diversity. Where there's no real effect of fences on light penetration, we don't see as much of an effect on diversity loss. And so it's a directional resource that the herbivores are controlling here. And we don't see a difference across control or fertilized. It really has to do with that change in light, regardless of whether it's fertilized or not fertilized. Interestingly, the same thing is happening here. We're losing those native species differentially. The non-native species are across the board. No strong patterns here in their response. So it's the native species that are differentially being lost. Interestingly, this is work that was led by Jody Price, Judith Sitters, who's at NEO, and a whole lot of other non-net people. They came out relatively recently, looking at evolutionary history of grazing. And in fact, that loss of native species, that loss of species in general, tends to be happening at sites with a very long evolutionary history of grazing. Again, thinking about those native species needing to have that association with the herbivores, probably through this pathway of light. So both total diversity and native diversity. We see an effect in sites with long evolutionary history, but not in sites with short evolutionary history of grazing. Okay, so what? Bug people in the room. That's all nice about like the big herbivores and all, but what about the small ones? I wanted to understand more about growth and defense. So Eric Lind was a postdoc with me and looked at growth defense trade-offs. And what he found is that where nutrients increase the cover of certain species, those species also are the species that benefit from putting up a fence, so removing herbivores. Right, so we see turnover toward less defended species, but we also see this growth defense trade-off. So that emerges from this. Okay, so what? Does that matter for the consumers? Yeah, it does. So we see this ramifying to impact arthropod damage. Again, control NP and NP. This is a paper that Anna Abling, who's at IDIV in Germany, and Dana Blumenthal in Colorado led, showing that when we add nitrogen, we see greater leaf damage from the arthropods under the nitrogen added, right? They're yummier. And we see when we have biomass of arthropods, right? So we went out and we sucked up all the bugs in plots. We found that wherever we added N, we got greater biomass of arthropods. So again, this is a later paper that Eric Lind led. That was the one that we were talking about earlier. So in both of these studies, the nutrient fertilization and the fence by fertilization study, we found that same effect. Okay, so I told you a lot of different pathways through this talk that could be arising, right? These are both observed variables, the biomass of the arthropods and well, everything to the right of the treatments themselves. So we tried to pull this apart using structural equation modeling. And what Eric found with this model was that plant structure, so the mass of plants was the best surrogate he could use for structure, impacted arthropod body size. But there was an additional variable of plant quality, the chemistry, right? So there are two pathways that are leading to this change in arthropod body size, and ultimately to secondary production. So there's a turnover, that compositional turnover of arthropods is moving toward larger bodied organisms as well. So it's a very metabolic type of story that's emerging there. Okay, I've regaled you with all kinds of cool data, and there's so much more I'd love to tell you about, but I ran out of time. So what I take from this is that yes, there are mean effects, and I showed you some of those, but there's also a more subtle, regional, climatic, plant compositional effects that arise and that we can start to quantify. And what's controlling the vegetation, diversity, productivity, and other characteristics that flow through these paths, but we can start to quantify those relationships and determine, will this be a dominant force under high precipitation? Maybe not, but it will under low precipitation, for example. And so stepping back, I think about this banding pattern on Earth, and that's what sort of motivates me in my own science, is understanding where will vegetation be? It's our battery, it's our sunlight storage on Earth. Where will it be and what will it look like? How will it function under future conditions? And at first pass, maybe climate, we're done. But I think what we're doing here is shedding some really interesting light on these other factors. And this is my other field photo, so it's again a little bit hard to see. But when I took undergrad ecology about 2 billion years ago, well, not quite, we learned that at high latitudes, vegetation is controlled by short seasons, cold temperatures, it's climatic, right? So this is an image from the tundra in Finland. And this was sent to me by Anu Eskalainen. And both of us were just agog at this picture. So they put up fences to keep out caribou. And suddenly they had what looks to me like a British lawn. It's got a dandelion. I mean, really, like that is not strictly controlled by climate. That fencing that they hadn't done there before, right? They didn't know, right? That tells me that there are other very important controls in these regions, even when we have the assumption that, well, climate will answer the question. If we have changing relationships with herbivores and fertilizer, whatever, elements coming into our ecosystems, we may get the answer fundamentally wrong if we don't include that. And so stepping back here, that was more or less what I wanted to say, is that yes, we at a first cut can model the world with MODIS or whatever we might do, looking just at climate and color, but we're going to miss a lot of really important future prediction without including other relationships. And so through collaboration with people all over the world, we're able to start to get an understanding of where and why and when these relationships with the vegetation and herbivores will change. And so I need to thank a whole lot of people. Eric Seablum is my co-lead of this project. Also at NEO this year, and an awesome human. Peter Wilford, Ashley Asmus, Eric Lind, oh, Lydia. For those of you online, if you can see Lydia, she's great. These have been the data managers, postdocs extraordinaire who have helped to wrangle data and people through the past 15 years of this project. This is just one example. I had to put a picture with Enrique. And Pedro up at the top a while ago. This is just one group of scientists that's come together at Minnesota, but each year we've had meetings of the scientists, and the people are really what generate the ideas and keep this network together. We've gotten funding from lots of places, thanks. Keep it coming. And with that, I'm done. Thank you.
I'm sure there's a question. How does this work with the microphone? Are these the outlets?
Yeah.
So,
Yes. Stop.
please, go ahead. Questions? Not all together, please.
Or not.
Yes, Anita.
I have a question. I have many, but maybe I'll start with one. I mean, there's so much literature saying that herbivores promote plant diversity. Were you surprised that you didn't find this overall effect?
Yeah, I was a little bit stressed, actually. The first time I made that plot, I was like, oh my gosh, how am I gonna explain that? But that was what I set out to test, was these covariates, right? There is no I have not seen a meta-analysis that has had consistently measured light, for example, but it's a critical resource and we are missing that. And so that was the hypothesis I specifically wanted to test. So yes, overall, but at the same time, stepping back to that earlier slide of the guinea pigs and the news and the wildebeest around the world, that was, they don't all have the same impact on diversity in their different systems. And so the layers of understanding of, evolutionary history is important and light is important. And so being able to add those layers on to say when and where that would matter has been super exciting to me. So I think a lot of that literature comes from grazing lands and comes from rangeland work where there's a different relationship between the herbivores. They're subsidized, and we did see that effect. So, yeah.
Cool.
Just picking up on the evolutionary history, I guess from a plant perspective, I don't know. I get very confused because 1.5 still doesn't seem like, or whatever percent you have, doesn't seem like very much to me.
1.5 what? Sorry, which?
Percent of the plants.
Plant species.
I realize it's a ton of species, but I'm always surprised on how many plants there are on Earth.
Yeah, exactly. There are a lot of plants on Earth.
But I mean, so if you're looking at evolutionary history, maybe from the plant perspective, it's hard to start to make generalizations.
Yep.
But maybe from the herbivore side, as you sort of point out, you have big groups of herbivores and you can start to ask whether certain, groups result in, or maybe it's just body size, but result in certain kinds of patterns.
So the Price and Sitters paper was specifically looking at regions that had an evolutionary history with hooved grazers.
Okay.
But that was looking at the region and not the species. So now one direction that there's a postdoc who's heading down this direction, looking at the phylogenetic history of the plants themselves, because these are all, assembled, not necessarily intentionally assembled, but a lot of these grasslands have anthropogenic fingerprints on them. And so there are plants that are native and non-native. And so looking at the evolutionary history of where they came from evolutionarily to start to parse apart some of that lineage. And no, we don't have the world of species, but yes, we actually have pretty good distribution across the phylogenetic lineages of plants. And so again, there are lots of gaps, but we can ask, are there certain lineages that respond in really different ways? So.
Yeah, nice. No, I was just wondering, because to me, it's such a big challenge because there's so many plants and yet deep down inside, of course, evolutionary history matters, but how do you really get at it?
Yeah, that's right. And so each of these is like a nugget. We're like, mostly what I talked about, sure, yeah, I showed you a bunch of graphs of published stuff, but behind every one of those graphs is like 50 new questions. Of course. And so that's among them with that evolutionary history of grazing paper. It's like, but wait, There's so many other, like the plants themselves in those locations might not have had that same history.
Yep, nice. Thank you.
Yeah.
Right. Oh, yeah. Yeah, thanks for the talk. Yeah, another question maybe. I was wondering if you look at how available this nutrient that you added in the soil were to the plants or to the microbes or to the system.
Yeah, already.
Yeah, that's that would be critical, I think.
Yeah, so availability right now, the chemistry that we have through time, we haven't done availability, we've done total. So example, total nitrogen, total carbon. But there has been work looking at different fractions of carbon, for example, which have strong associations with nutrient availability. There's work that Anita can tell you about looking at some of the rates of nutrient cycling as well. So yes, there's ongoing work. We had a resin bag study that was done across sites that ended up not producing data for sad reasons. We all have those projects, right? And so we're hoping to do a resin bead study into the future to look at, try to capture more of the available nutrients. So yes. Any other ideas for cheap ways to capture nutrient availability around the world? I'm all ears.
And the bacteria get lazy.
Yeah, right. We were talking about that.
They stop.
Yeah, well, that's a whole other story. I mean, yeah.
Yeah, exactly.
What is below ground? What's
Oh,
happening there? I
Well,
mean,
that's a whole other talk,
yeah.
which I haven't written yet, but there's we have a lot of data there as well, and actually increasing data on soils. We're doing a big metagenomics study right now, looking at functional groups of microbes in the soil.
Cool.
So, TBD.
And it's difficult above ground, but it's more difficult below.
Right. Yeah. One of the things that we've had to balance is wanting to have a very long-term study, wanting to study below ground processes and not wanting to just, I don't know, not Swiss cheese the plots, right? Not leave holes so that we don't know what the soil, so trying to keep the soils intact. So that's been a challenge, a balancing challenge.
Yes, I would have another one. Well, vertebrate herbivores are expected to be less specific and probably act on plant diversity by affecting via this growth defense trade-off. And I was wondering, what do you expect for invertebrates or pathogens which are more, where there are more specialists and which also act on diversity via negative density dependence? Would you expect stronger effects on plant diversity, or given that they might not affect biomass so much?
I guess what I would say from our, taking our result and turning it around is the arthropods were able to get to all of the plots. And so despite that, we saw effects across fence lines. So they're at least not negating the effect of the vertebrates. I don't know about, I can't separate out condition-specific results, but at least across the data set, the vertebrate effects were there. That said, I would love to insecticide plants. I mean, yes and no. To understand more about the interactions between vertebrate and invertebrate, because, invertebrates can change the chemistry of the plants too through their consumption. So we do have the data from Anna and Dana, that paper that I showed you, where we did see increased damage. I don't know. It's speculation to think about that. But I guess your comment about the vertebrates, I think one of the things that I've started thinking about a lot in these plots is that we have vertebrates, but I don't think that in a lot of sites their major function is consumption. I actually think that I have deer that bed down in my plots and they smash the vegetation down, which means that the litter is right next to the soil. And so I think decomposition processes are speeded, but I think that's not because the deer I mean, they like leaves off of woody plants as much as they like grassland plants. And so I think one of their major effects is non-trophic,
That's interesting.
which I want to, yeah, I haven't figured out how to parse that apart. But body size might be one of them. And I don't know if we can do personalities, tendencies of herbivores to do certain things in plots. But the high biomass plots also tend to be really good cover for their babies. And so they'll bed down in those. Anyway, it's, yeah. And there's the whole below ground vertebrates. Yeah.
Another question?
That's okay.
I was wondering, first, thanks for the talk. So thinking about, for example, South America, there are nowadays a lot of, like from a historic perspective, a lot of big herbivores got extinct. And do you think there will be like an effect on the sites that you're having a look at, like grasslands where the diversity of like also functional diversity of herbivores got lost quite a lot in the past? Would this change?
So you're asking if the evolutionary history,
Yeah.
well, I mean, we have some evidence the evolutionary history does matter. We haven't taken it, that's one of the questions behind the slide.
No.
We haven't taken it to the point of asking, if we restore them to some extent, I mean, there's the whole rewilding idea. If we have cattle that can serve in that same function, or if they're functionally completely different than the herbivores that were there previously, what do we see in terms of the response of diversity or biomass? But we also have low replication in terms of domestic herbivores at our sites. We just don't have a lot of sites where we have domestic herbivores, so it's harder to answer that one in this network. But yeah, there are definitely different effects in different locations, we know that, and so understanding more about that is maybe for a different study, I don't know. Maybe we should do it, maybe a meta-analysis would be the next step for that, to just see what's out there.
No? Very good, thank you. If there is not a pressing question, it's already 12 and probably people are hungry. But whoever wants to join us for lunch, I reserve the table. So please feel happy to join. And thank you again, Elizabeth. Thank you guys for coming. Thanks to online people for being here.